This is a water bear. It may look scary, but it's actually quite small. Water bears are typically around 1/2 millimeter in length. This image was taken with a scanning electron microscope and then colorized, so that's why it looks kind of three-dimensional. This image that we took through a regular light microscope is what a water bear typically would look like to you as an observer. This water bear was collected in some moss that lives locally, but water bears can be found all over the earth. In fact, they're known to be one of the hardiest animals to exist. It's thought that water bears can even exist in space for some period of time. We decided to test that hypothesis. So what we did is we sent up a high altitude balloon. Now these high altitude balloons go to the edge of space, and we sent a water bear up in it. What we did is we collected some moss from this roof here, and we kept some moss down on Earth as a control group while we sent the other moss up to the edge of space in the weather balloon. When they came back down, we looked at them in water, gave them a chance to hydrate so we could see if the ones that went to space survived, while seeing if the ones on Earth also survived as a control group. So we collected this moss from this roof, kept half of it on Earth. The other half we attached to this lid of a milk jug on the inside of the lid with hot glue. And that lid then became the top of our spacecraft, this foam box. The spacecraft just contains a GPS so we can find it when it lands, and some weather instruments like a thermometer and a barometer. This picture was taken at the top of the high altitude balloon's flight. You can see the sun and a black sky. That tells us that the balloon is above almost all the atmosphere. In fact, the balloon is above over 99% of the atmosphere. This graph shows you the air pressure over the course of the balloon's flight. You can see down near the bottom of the flight at ground level, the air pressure was quite high. And as we went up in altitude, the air pressure got lower and lower until it got almost to zero. Air pressure is a measure of how much air is above you. It's a measure of the weight of the air. So an air pressure of 0 would tell you that there's no air above you. That's how we know that we were at the edge of space. So this is where the water bears were. They landed about 20 miles away from where they took off from, and we collected them and put them in water to find out if they would rehydrate and survive the flight. Our mission to the edge of space is over, and we've got our moss samples. So these two moss samples went to the edge of space on our spacecraft, and these two moss samples, both from the same location, which was my neighbor's roof, by the way, did not. They stayed here the whole time. So what we're going to do now is we are going to put these two moss samples that stayed here on Earth in this beaker. And then we'll get these moss samples. We actually hot glued them to the top of this milk jug, by the way. And the milk jug's purpose was to keep our spacecraft right side up when it landed. We taped it to the top. And since it comes to a point in the spacecraft, if it rolled over like this, it would tip back like this and be on its side, make sure we get a good signal. So that's why that went, but it served a double purpose of protecting the moss on its flight. And yet, because it wasn't sealed, this was open, the moss was exposed to the vacuum of space. So now we'll add some water to each. We're using distilled water because we know that water bears and nematodes and rotifers don't survive well in tap water. So there you go. We're going to let these sit for a couple of days, and that'll give the moss a chance to hydrate, and the water bears and nematodes, if there are any survivors, a chance to come out of their dormant stage and be crawling around, and then we will look at them, both samples under a dissecting microscope, and we'll see if we see life. We'll be able to answer your hypothesis regarding whether or not water bears can survive a trip to the edge of space. It's been 2 days of the moss sitting in distilled water in these beakers. So now what we're going to do is, this was a beaker here with the moss that stayed on Earth. So we're going to take the moss out. I'm going to squeeze it out over this petri dish. Try to get any sort of life that's in that moss kind of squeezed out into there. I'm going to go wash my hands so I don't contaminate the other moss with life that from the moss that stayed on Earth. And I'll come back and do the same for the other moss. All right, so I've cleaned my hand off, and now I'm getting the moss that went to the edge of space. Squeeze this moss out over the petri dish. And now we'll take a look at each of these under the dissecting microscope and see if we can see anything. So here's our sample from that stayed here on Earth. And we have quite a bit of life in this one moving around in there. And I think that what we have is rotifers. I don't see any water bears in this sample. However, I do see a lot of I guess we could call them biocysts, which is the dried up form of these microorganisms. So I feel like even though it's been 2 days of sitting, there's still some life that has to fully rehydrate. Here we see under higher magnification, 100 times magnification, from our sample that stayed on Earth, some rotifers. You can see them causing little currents in the water around them with their rotating parts. And what that does, oh, that one just ate a little bit. What that does is it sweeps tiny little food particles into its gut. These are about 0.5 millimeter long. So this is an actively feeding rotifer. Those other little blobs there that are kind of the same color but aren't moving are rotifers that are not fully hydrated yet. So I think we'll come back in another day, see what else is living in here. So here we have the view in the microscope under 40 times magnification of our moss that went to space. And you can see there's life in there. You can see stuff moving around. So stuff survived that trip, but we don't see any water bears. What we do see is rotifers. Those main things that are moving around in there are called rotifers. So that means that these rotifers can survive the trip to space, at least briefly. Here we see in our sample that went to the edge of space a number of rotifers crawling around. It seems as though there's more rotifers in this sample than there was in the sample that stayed to Earth, but that could be because it's of something about our experiment, or it could be because just this clump of moss had more rotifers to start. We really don't know. You have to have a larger sample size to say with any confidence. But there you can see a few active rotifers, and then the things that look the same color but aren't moving are rotifers that aren't hydrated yet. Or possibly water bears. I'll have to come back in another day or so and see if any new life is rehydrated. So we just experienced one of the great things about science. We set out to find out if their water bears could survive the journey to the edge of space or not. And what we found out instead is that our moth samples didn't have any water bears at all. However, we did find out that rotifers could survive the journey. We found rotifers both in the sample that stayed on Earth and rotifers in the sample that went to the edge of space. We have experimented with this moss before, and we found that the rotifers rehydrated quicker than the water bears. So maybe we'll come back in a few days and see if we see water bears in either sample. We did find that the rotifers can survive the journey to space, and that adds another species, or at least a genus, of organism that can survive at the edge of space.